Hi, this is Dr. Ben Morrill. Welcome to episode 88 of Reptile Genetics Weekly. Glad to have you here. And today we're going to talk specifically about the nuts and bolts of how the Ultramel morph works in ball pythons. We talked briefly about it before, but in this one we're going to show, what's changing in the code and the protein, and talk a little bit about specifics that we've learned that you all appreciate. So we're going to hit that much deeper than we did the brief time we talked about it before. As far as updates, we have fast tests going out all the time. We've got sex determination going out a couple times a week. For the panel tests, this is going to be a really big run with lots of research on it. So it's taking a little longer, a couple days longer than I thought last week when I talked about it. So I would say we should have results late this week into the weekend and through till Monday. It might take us till Monday to have all of the results analyzed and sent out to everybody. So it's got a little bit of time, but by next week, when the next episode we have up, I'll be able to say all those results are out. They should be. And that also means I'll be able to look at this big research run, and so you'll be able to tell by how big the smile is on my face how many of those look like they're doing well. And hopefully we can reach our goal of getting to 55 morph tests by the end of this month. This will be a big part towards that, so we'll see. But yeah, I think that's all I have for updates. Kayla, how are you doing?
Hey Ben, I'm doing well. I'm looking forward to getting into the weeds with these genes, and we're kind of the perfect place for it. And I mean, it makes sense. We do reptile genetics here. It's Reptile Genetics Weekly. But it just makes me really happy that our audience has really responded to when we get off into the weeds about like what causes these proteins to do what they do to change these colors. So it's really cool. And Oh, and all my snakes are officially down in brumation now. So I have far fewer snakes in my life right now than I would enjoy. But it's fine. They're literally right next to my bed because that wall gets really cold.
Oh, there you go.
Yeah, so I'm just like, we're just gonna stick them right in there. But it's good. But yeah, let's start off with some business stuff real quick. For those of you who haven't used it yet, we definitely recommend checking out our a la carte style testing or custom bundle shed testing through our vendors, Clutch and Morph Market, whichever one you go for. With morph testing for ball pythons, you can bundle them to save some money. So we highly recommend it. It's a way to get you more testing for less.
Yeah, so if you have animals that are pos triple hets or something like that, or it's possible double het for a couple things, or maybe it could have yellow belly or not, could have this or that. This is a really good way. It's not going to cost you very much more to add a couple more on or whatever. So we're very glad that Clutch and Morph Market programmed that in, so you all have that opportunity. You can customize your own 5-gene panel as you would.
As you do. And I mean, it's nice because we kind of, at least with ball pythons, you kind of live in a quad het world now, a lot, or at least they're a lot easier to come by than other species. So it's definitely worth it to prove those out even before the animal is even close to breeding size. But yeah, let's talk about our monthly-ish morph spotlight. This time with Ultramel. We got a nice picture of a yellow belly spot nose Ultramel from Canova. Just really pretty animal and a nice little example of the gene we'll be talking about the most today.
So for this, we're going back to the second paper. So the piebald was the very, very first one that was published. The second paper was with Dr. Hannah Siddell and her students at Eastern Michigan University. We've talked about many times. So thankful for the work that they did so early on, and very, very cool that Dr. Siddell made it possible that students that are taking her class got to take part in an active research project that affected us in our industry. Her students got to be a part of that while they were in her class. So very, you don't see that very often. So
no, not everybody gets out of undergrad having already, having their name in a paper. That's really cool.
Oh, so yeah, in this paper, as we talked about before, this is the same paper they talked about lavender albino and then also albino. And that's one that we only kind of touched on really briefly. So we'll come back and talk about the specific mutations for albino and candy toffee. We'll do that in another episode. So you have albino in this paper, you have lavender albino, and you have Ultramel. So very, very cool work.
Yeah. And I love how, like it just in the opening abstract, they say color morphs in ball pythons provide a unique and largely untapped resource for understanding the genetics of coloration in reptiles. And that makes me really happy that, well, I mean, of course, Dr. Seidel is familiar with the ball python community, especially after working with all of the people who have contributed sheds to her work. But it's nice to see, a little bit more of the reptile hobby and the science world kind of holding hands a little bit more, and recognizing the untapped potential of working together. So that's really cool, I think.
Yeah, that's something that was frustrating to me early on, knowing that breeders had looked down upon the researchers, and then the researchers looked down upon the breeders, and I
Right.
was like, we can both learn from each other.
You
So
are literally the only guy I know who has a doctorate in ball python genetics. So yeah.
yeah, there's not very many people that have done a master's or PhD on ball python genetics. That's for
Yeah.
sure.
Yeah. All
So
right.
yeah, in this picture, what they're talking about is the kind of continuation from in A. There you have a normal, colored ball python,
Mhm.
and as you go to the right, you get less and less melanin. So
Mhm.
melanin is the dark pigmentation, and all the way on the right, that's that albino. That's where you have no melanin at all. So you go from normal on the left in A to completely no melanin in D on the right. And so as you move across that, if you take a little bit of the melanin out, then you get ultramel. You take a little bit more out, you get lavender albino. Then you just take everything out and you get an albino. So
Yeah.
that's a pretty cool figure. I was glad that they put that in there. And then they have some of the genes that they thought might be playing a role. So
Mhm.
this is before they started studying these. These were kind of the ones they thought may play a part in these different, causing these different morphs. And then they, of course, studied and found out what they actually were.
Yeah.
And Thor just ran up. He wants to say hi.
Oh my gosh. Hi,
Hi,
Thor.
Thor.
Oh, what a good boy.
All
He's such a good dog.
right. He's supposed to be going outside, but he ran directly to me.
Oh, he had to say hi to his dad, of course.
I guess. All right. So this first table here figure is interesting because as we've talked about how we design these tests, and we
Mhm.
sequence, in this case it'd be a bunch of ultramels, and then we sequence a bunch that we know don't have ultramel, and then we take all of that sequence and we ask the software to tell us where the software sees a change in just the ultramels, and that change is not present in the non-ultramels. And
Mm.
any of those changes that are unique to ultramel animals, then we start searching to say, okay, which one of these is the actual ultramel mutation that we can design a test for. Well,
Yeah.
one of the things that you can do once you have a possible mutation you think might be causing this morph, this look, you could look and see how important is that specific part of the code. So some parts of the code don't code for proteins at all. Other parts code for proteins that are super important. And if you don't have it, it'll kill the animal. And both of those ends of the spectrum we're not really interested in, because if it's such a bad mutation that kills the embryo, then we know that's not what causes ultramel, because they wouldn't be born. And
Mhm.
if it's one that doesn't have anything to do with coding at all or expression, then we already know that, okay, then that's not going to be what causes ultramel, because it doesn't code for anything. So this is a really interesting one, because what we see here is when you go across elephant shark, zebrafish, clawed frog, human, chicken, painted turtle, anole, Burmese, ball python, all of those different, obviously fairly unrelated species, this specific place, this 305th codon, which so 3 bases, A's, T's, C's, and G's. Each 3 of those will code for 1 amino acid. And so when you're looking at the amino acid sequence, 305th is this R, and it's in all of those different species. So that's you can see in white, some of the other positions, there's some changes in different species. But for that R, it's there across all those different species. And that's a sign as a biologist, when you're molecular biologist looking at this, you're like, okay, if across all those very unrelated species, if that's an R in all of them, that's probably pretty important. So that must have a significant role in whatever that protein does. It might be what binds, like when we talked about hypo ghost and hypo ball pythons, we talked about the reason why they become hypomelanistic is because of the protein that transfers that melanin out gets messed up, and so you're not able to move that melanin. Still gets made, but it doesn't get transferred out to where you can see it in the ball python after it's born and stuff. So in this case, where this R is, that could be playing an important role for that protein, and specifically for this gene, which is the TYRP1 gene. That's in helping to make melanin. So with hypo, it was transporting it. With Ultramel, it's in making the melanin. So you have this reduction because this R gets changed. It's not able to produce melanin as efficiently, and you get that reduction in melanin that gives you that Ultramel look.
That's so interesting that the hypo mutation technically produces more melanin than this. It just doesn't get moved around as much. Whereas this one, it is actually not producing as much melanin in the first place.
Yeah, so it's a different part of the process that gets messed up by that.
Yep.
So yeah, in this figure, what they're showing is, so this, unlike lavender albino, where every lavender albino they tested, they saw the mutation that the animal was homozygous for it. So they found a mutation they thought caused lavender albino, which was a deletion. Made a test for it, and then every lavender albino they knew visually was a lavender albino. Everyone they tested was homozygous for that one mutation. And so that's one mutation, one morph. But what happened here, this figure kind of tells a story. They sequenced one Ultramel animal, and they found it had that R changed to an H. It was homozygous for that change. And so then when they went and got a bunch of other Ultramels that were visually Ultramels and tested them, some did test homozygous for that mutation, some tested heterozygous for it, and some tested negative for it, even though they looked like Ultramels. So They knew visually they were Ultramels, but that one mutation didn't tell the whole story. There had to be something else going on that could also cause an Ultramel. And so what they show here is they had 6 animals that were homozygous for that R305 change to an H. So that's what that R305H means, at position 305 in the amino acid sequence, an R changed to an H. And so then the other mutation that they found later was in the coding regions 6 and 7. So it was further on in the protein or in the gene sequence. If you think about it in DNA, it's in the gene sequence. For the protein, it's the latter part that codes for the end of the protein. And so in 2 of the animals, they only got heterozygous for that R changed to an H. And then once they knew about the other larger change towards the end in exon 6 and 7, they found out that 2 of those animals that were heterozygous for the R changed to an H were also heterozygous for that deletion towards the end of the sequence. And then 3 of the animals were homozygous for that change at the end. So in the end, what we have, as we've communicated to you all, Ultramel A is this R changed to an H. It's a SNP. It's one little change. So when they were originally doing this, they would have gotten Ultramel A tests. They would have gotten 6 of them correct because they were homozygous for that.
Mhm.
they would have gotten 2 wrong by calling them hets, and they would have got 3 others wrong by calling them negative if they were only testing for Ultramel A,
Yeah.
because the 2 that tested het for Ultramel A were also het for Ultramel B, and then the 3 that would have tested negative for A, that's because they're homozygous for B. And so that's kind of a fun part of the process, is they were figuring out what was going on, and they knew pretty quick that it can't just be one mutation doing this, because they have obvious Ultramel animals that are negative or heterozygous for the first mutation they found, and thankfully they were able to find that second area towards the end of the coding sequence that explains how we get Ultramels that have potentially 2 different mutations going on. The
Mhm.
last number there, that's 78. Those are non-Ultramels, so those are animals they expected to not be Ultramel at all, and they did not have the R changed to an H, and they also did not have that larger deletion or mutation area towards the end of the coding sequence.
Mhm.
So all 78 of them they expected to be negative did not. That's an important thing, because if we found out, oh, out of those 78, 5 of them have that SNP, that R changed to an H, then we would know, okay, that doesn't actually cause Ultramel. But the fact that the 11 that they looked at either had one or the other or both of those mutations, and then 78, quite a few animals that they expected to not be related to Ultramels at all, those 78 were all negative for both. So that's another really important part. And this figure here just shows the like we've talked briefly about this before, for this TYRP1 gene. It's starting on the left. That arrow, that's the promoter, kind of signifying promoter or start site, where that gene starts in the DNA code. And as you go to the right, you can see on the I guess that's the 4th codon, or the 4th exon, I mean.
Mhm.
You have that R305H. So that's where that mutation is kind of in the middle of the sequence for the gene. And then if you move down to the next one, you can see at the very end on the far right is where you have that. At the time, they didn't know for sure what that more complicated, larger mutation was. They just knew it was towards the end, and it was likely over 2,200 base pairs that were changed. And to date, so when this publication was made, Dr. Siddell did not know what that was exactly, but
Mhm.
since then she did figure it out and helped make it so we could test for Ultramel B, and so that's what made it possible for us testing companies to be able to do that, is because she did figure that out, but that's just not published yet.
Okay. Can I ask, just from a general layman
Yeah.
uh view, like when you read these type of uh what is this kind of graph called? And like, you've explained what the exons are, those little peaks there, and the R305H, that's the same as this right here,
Yep.
representing all of those amino acids.
Yeah, so
Um,
this here is an amino acid.
uh-huh.
Yeah, this is an amino acid sequence that you're seeing here.
Yep.
And then this picture here, this is showing the DNA code. And so there's
Okay.
spaces in between the code. So the exons are the boxes, the black boxes.
Mhm.
And then the lines in between, those are introns or non-coding regions.
Oh.
So all of that has A's, C's, T's, and G's, but the ones where the black boxes are, those are the A's, C's, T's, and G's that matter for making the actual protein. The actual TYRP1 protein
Mhm.
is really only coded. We're reading from left to right,
Mhm.
and it's going to read what's in the box, and then the code tells it to then skip until it gets to the second box. So
Huh.
there's DNA code, there's A's, T's, C's, and G's, and sometimes it's like 20,000 base pairs or whatever. There's a whole bunch of code there, but what's making the protein, it knows to skip all of that, go to the second exon and
Mhm.
read that and add that to the protein, and skip to the third one, read that, skip to the fourth one. And so even though there's DNA all along that whole thing, it really only matters, and it's going to read and use the code that's signified by the boxes to make the actual amino acids that then towards the end of at the end of it, when it's all done, it ends up
Mhm.
being protein.
Okay. Thank you. And so So the lines, or what did you call the lines themselves in
They're
between the exons?
intronic or
Intron.
non-coding.
So the non-coding material is what was previously known as like junk DNA, right?
Yeah,
Even though that we're finding that actually the junk DNA does have its uses.
some of
But
them.
for our purposes,
Yeah,
yeah,
yeah.
okay.
Some of it we still don't know. There's still parts of it that we don't know what it does, but
Mhm.
we're more hesitant to call it junk now because
Right.
there's a lot of stuff we thought wasn't doing anything, and now we know it is doing something. So
Fair enough. Okay. Well, thank you for explaining that. I'm sure other people in the audience probably had similar ones. So, appreciate that.
oh,
So yeah, um, here is here's our Ultramel A and Ultramel B.
yeah, so they are, as you saw in the last figure, they're fairly close to each other. They're just in different parts of the gene.
Mhm.
But when you look at a chromosome, on the gene, they might be, 10 or 20 or 50,000 base pairs apart. But that whole chromosome itself is like a lot of the time 100 million base pairs. So when
Yeah.
you're looking at it at that scale, 100 million, when it's 10, 20, 50,000 base pairs apart, that means they're stacked on top of each other. They're right by each other.
Mhm.
So yeah, Ultramel A, Ultramel B, they're right by each other. They are in the same gene, like we just talked about. A is a SNP. That's the R305H mutation that we were talking about.
Mhm.
And then Ultramel B is that larger mutation towards the end of the gene that to date has not been published yet, but I would imagine at some point Dr. Siddell will publish that as well.
Exciting. So, as far as the test itself, the morph test that you get when you order a test for your snake, we grade this as an A plus.
For Ultramel A, yes. So I guess overall,
Yeah,
because
sorry.
every time you send in an Ultramel test, we're gonna do two tests on it. We're gonna run
Mhm.
A and B. And so I guess overall, we would have to say the Ultramel test is an A and not an A plus, because the reason why we give it a plus is when it has a scientific publication behind it. And technically, Ultramel B doesn't have that yet. The details, the specifics for that larger mutation towards the end of the coding sequence.
Mhm.
So I guess you have to overall give it an A.
Yeah.
But yeah, the first one is very, very well. They've both been run by us and other testing companies and Dr. Seidel, and many, 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 many times by this point. It's a very, very
Yes.
good test. But it is important to remember each time you do that, we are needing to run two different tests, just like we have
Mhm.
to do for albino and we have to do for yellow belly. And
So Ultramel A is the A plus because it has a publication with it, whereas Ultramel B works great for us, but there is not a specific publication for just the Ultramel B test. But since we've run it enough, we know it's a solid test. We consider it an A, which is pretty darn good. And here's a buttload of names. These are the people who have contributed sheds for this study, and so we consider all of these people as this week's shed donor shout out.
yeah, big
And
shout out this time.
yes, definitely a lot of familiar names in there.
Oh yeah.
And even at the bottom, people who contributed images of the color morphs used in this study as well. I threw those guys in there because that's uh, actually a lot of them were also people who contributed the sheds, but we figured that is, I mean, showing the beautiful visuals of these animals that we work with. I think that's all important. So
Oh yeah.
um, we really appreciate all the work of all of these people. And I know Dr. Seidel and all of her students did as well, because they did
Yeah.
amazing work.
Yes, just like we always say, we literally cannot do this without you all helping, and they did a very good job of I think kind of opening people's eyes in the scientific community to the fact that you can get both them and the Canadian group. They talked in their papers about the fact that they're able to get these samples to be able to do projects like this just sent in from people that keep these animals, and that's really, really cool. Like we talked about earlier in the episode, awesome to see keepers and breeders be able to be a part of scientific research.
Yeah. I mean, at the end of the day, you basically have a huge untapped, mostly untapped hobby full of genetics nerds who are just doing the breeding on the ground here. And
Yep.
you know, there's so much studying you can do on this. So definitely cool to see it, and we really hope you guys enjoyed you know, looking under the hood of the Ultramel gene today.
Yeah.
So yeah, thank you, Ben, for explaining it to us today. And for those of you, if you enjoyed it, definitely drop a comment, subscribe, like the video. I'm also going to link some of our other morph spotlights in there, especially the hypo one. If you want to know what I meant about, you know, the hypo gene creating more melanin, just not moving it around. So check it out. It's definitely worth it if you are a genetics nerd like us. So thanks, guys. And yeah, got anything else, Ben?
Nope. Just let us know in the comments if you have any questions, and like Kayla said, please do subscribe and like. If there's something that we're talking about you like, let us know, and let YouTube know so they'll share our videos with more people.
Yes, please. Okay. All right, guys. See you next week. Outro in 3, 2, 1.